So here we're going to do a little bit of probability. The probability can be used to predict the results of crosses, one trait or two trait crosses. So you're learning outcomes here to explain the rule of addition and the rule of multiplication and apply these rules of probability to genetic crosses. So I'll give you a couple of examples using the rules of probability to solve even more complex problems than we've seen so far. So probability is going to allow us to predict the likelihood. Doesn't mean you're going to always get exactly what you get there, but it'll give you what are the chances. So what are the chances of flipping a quarter and getting heads? Well, it's 50-50, right? You get 50 heads, 50 tails, 1/2 chance or 1/2 chance, right? But you can flip a quarter 10 times and just by random chance you get all heads. It can happen, but if you keep flipping over a large number of flips, you'll eventually start getting closer and closer to what the true probabilities are, getting closer and closer to 50-50 or 1/2 and 1/2. So the first rule we're going to look at here is the rule of addition. And the reason, by the way, why we can use probability is because, first of all, in diploid species like humans and plants and other animals, our body cells carry diploid number of chromosomes, and we have two copies of every gene because each chromosome has one copy of those genes. And so the law of segregation that we learned in a prior section says that the offspring is either going to get one copy or the other from one of the parents, and the same is true for the other parents, going to give one or the other. So we're going to split, and the copy that you get is a random chance, just like flipping a quarter. So these are the reasons why we can use probability for this. And so when we look at the rule of addition, it states that the wording is confusing, but I'll try to explain that here. It says the probability of either of two or more mutually exclusive events occurring is the sum of their individual probabilities. Mutually exclusive means that if you're going to get an event to occur, say the fertilization of an egg, what is the probability of you getting a homozygous dominant? Two capital letters, right? Both alleles are dominant. Well, that depends on what you got from one parent, because you're going to get one from one parent, you're going to get one from the other. And once that happens, you can't get any other genotype, right? So that's exclusive and different from getting from the first parent, big A, and the second parent, little a. You're either going to get this or this. So these two events, those are mutually exclusive. You can't get them both in one individual, okay? And you think about it, we're talking about the first parent. The first parent could well have given you a little a, and the second parent the dominant allele, so the recessive allele from the first parent. So technically, both of these are heterozygote, but you got them in a different way, right? In the first case here, the first parent gave the dominant allele. In the second case, the opposite was true, right? So these are exclusive because the individual can't have this genotype and this one. You either have one or the other. That's it. Okay. And then the same is true if the individual ended up homozygous recessive, right? So now we think about a simple monohybrid cross. Remember, a monohybrid cross, both individuals have to be heterozygote. Okay. And so when crossing them, it's asking what is the probability of producing an individual that is basically heterozygote. Okay. What is that probability? Now remember, there's two ways you can get a heterozygote. We mentioned them here. The first parent can give you the first parent. So let's say this is the first parent using P's here. The first parent can give you the dominant allele, and the second parent can give you the recessive allele. Or the opposite is true. The first parent instead gives you the recessive allele, and the second parent gives you the dominant allele. So there's actually two different ways, and they're exclusive of each other, that you can get that. Now, if we go back to our Punnett square, and we run this dihybrid cross, because we're we're doing the heterozygotes for both, and we separate the alleles, law of segregation. And we go and we run here. Remember, the Punnett square is a way for us to figure out actually what the outcomes, probability of the outcomes are going to be, okay, using the Punnett square. Technically, this one we might have written as little p, big P, is still heterozygote, okay? So we go with the first parent here, then we go little p, big P. Now, I encourage you in the prior section to always write the capital letters first, it's easy to see, but this is one way to get heterozygote. This is another way, and the probabilities of getting this way is 1/4, one out of those 4 boxes that are there, because these are showing what are the chances, right? And this is 1/4. So these are two mutually exclusive ways of getting heterozygote. So we apply the rule of addition because we can get it this way or this way. And if we do, that's going to be 1/4 plus 1/4, and that gives us 1/2 chance of being heterozygote. Okay, and that's what we were doing earlier. We might ask another question: what are the chances of getting the dominant, expressing the dominant trait? You can express the dominant trait if you're heterozygote or if you're homozygous dominant. So that's the first 2 squares and then the first one on the second row. So that's 3 out of the 4. So it's a 3/4 chance of dominant. And these are mutually exclusive, the offspring being homozygous dominant, because you get both dominants from each parent. Or first parent gives you the recessive and the second parent gives you the dominant, or vice versa, right? So you would apply the rule of addition there. And we were already doing that when we were trying to figure out the phenotypic ratios, right? So the phenotypic ratios, 3 out of 4 would show dominance, and then this last one here, both recessive would show the recessive trait, okay? So that's the rule of addition. Remember, when it applies here to genetics, we're talking about when you produce an offspring, that's an event, right? And you can't get both homozygous and heterozygous at the same time. You're either homozygous or heterozygous, and there's 2 ways to get that, and that's why we add the probabilities of those 2 ways, and we did that in the Punnett square. Okay. Now then there's the rule of multiplication. The rule of multiplication here, we're talking about independent events, okay? And the event of one happening does not influence the other one. And when you do that, you do the rule of multiplication. And so when we look at producing an offspring, okay, the offspring has to inherit 2 alleles, one from each parent. So we want to ask, what is the probability in this heterozygote, 2 heterozygotes, this monohybrid cross? And we ask, well, what is the probability of the individual being homozygous recessive? Well, the individual has to get the one allele from one parent, one from the other, and what the individual gets from one parent does not influence what the other parent gives them. So they're independent of each other. The allele you get from each parent is independent. So this is going to be multiplication rules. So we think back here, for this parent here, it's 50-50. You're either going to get the dominant allele or the recessive allele. That's 50-50. So it's a 1/2 chance to get the recessive allele, just like it's 1/2 chance to get the capital one, or the dominant one. And from the second parent over here, it's either the dominant or the recessive allele. It's 1/2 chance each, but we're asking about the recessive allele. And since getting from the first parent is independent of what you get from the second parent, we apply the multiplication rule. So 1/2 times 1/2 gives us a 1/4 chance. And so what is the probability in this cross? Without even doing a Punnett square, the probability of the individual getting this genotype is 1/4, which is the same as 0.25 or 25%. Okay? So we can approach it that way. What is the chance of the individual getting this particular genotype, right, from a monohybrid cross, where both are heterozygote? Well, one parent has one dominant and one recessive allele, and it's a 50-50 chance of getting each one. The same is true for the second parent, so it's a 1/2 chance of getting that one. So it's 1/2 times 1/2, and that would give you, again, 1/4, which is 0.25 or 25%. And if we think back to the Punnett square, that's what we were getting when we were doing these monohybrid crosses. Okay, without even doing the Punnett square, when we did the Punnett square, we got 1/4 one of the four squares, 1/4 chance of getting homozygous dominant. The other three squares didn't give you that particular one. Okay, so that's when you apply the multiplication rule, when you start thinking about what you get from each parent. Okay, so another way to look at this might be to use a tree diagram, and we think about the first parent. Okay, so in the first parent, if the first parent is heterozygote, well, the first parent is either going to give to its offspring when during meiosis and we segregate the alleles on the chromosomes, either going to give you dominant or recessive allele. Okay, and what are the chances of that? The chances of that is 50-50. So you can either write 1/2 or you can write 0.5. Your choice, right? Because 1 divided by 2 is 0.5. And then the same thing here, 1/2, okay, or 0.5. And then we look at, okay, let's say the first parent gives the dominant one. What about the second parent, right? Because we're crossing with the second parent, okay? The second parent, assuming the first parent gives you the dominant allele, what about the second parent? Well, the second parent also has one dominant, one recessive, right? And that is also 1/2 chance, and this is 1/2 chance. And in the second parent, these are the two possibilities, two possibilities that can happen. And what if the first parent had given the lowercase? Well, those are also. We're going to see the same thing. The second parent can either give a big P or a little p, dominant or recessive. So now what we do is we go along this tree and we multiply the probabilities and we combine what we get. If the first parent gives the dominant allele and the second parent gives the dominant allele, what are the chances of that happening? They're independent events, so it's 1/2 times 1/2, and so we're multiplying along the tree branches, right? And so the probability of getting big P or homozygous dominant is 1/2 times 1/2 is 1/4. And 1/4, by the way, 0.5 times 0.5 gives you 0.25. If we go along the second branch here at the end, the first parent gives you the dominant, the second parent has a 1/2 chance of giving the recessive, so you would get this genotype, and that's 1/2 times 1/2. You get 1/4, or 0.25. We can do the same thing for the bottom here. First parent gives you the recessive, second one gives you the dominant. Both of these, by the way, are heterozygote, right? And you get 1/4 or 0.25. This is just another way of looking at probabilities and how to calculate them with the tree diagram. And in the last case, first parent gives recessive, second one gives recessive, this is a possible outcome. And all of these events here are exclusive of each other, mutually exclusive, right? And you get 0.25. By the way, if you add all of these up, you should get 1 at the bottom. But we can ask several questions here. Okay? This is an exclusive event, exclusive. In other words, you can't get an offspring with both big P, and big P, little p. You either get one or the other. They're exclusive of each other. But we might ask the question, well, what are the chances of a monohybrid cross here? What are the chances that you get heterozygote? In order to do that, there's two ways. When there's more than one way to get the same, essentially get the same kind of outcome, heterozygote, you add. It's another way to think about it. So 0.25 plus 0.25, that would give you a 50% chance, or 1/2. You can write fraction or 0.5, or even, you can even write 50%, okay? We could ask another question. What are the chances of the individual showing the dominant trait? Because all you need is one dominant allele, then that would be these three exclusive events, and you would add those three, so the chances would be 0.75, 3/4, or you can write 75%. All of those are the same. And what are the chances of the individual expressing phenotypically the recessive? would be just this one here at the end, and that would be 0.25. Same as saying 1/4, same as saying 25%. Okay? So tree diagrams can be useful in that case here. You multiply along the tree, and then you add it depends on what question you're asking. If we're asking what is the heterozygote, if we're asking what is expressing dominant, then it's these three. Okay? So now that we have covered the rules of probability, we can see now that technically you would need to do a Punnett square. Remember, in a prior section we did a dihybrid. This is a dihybrid cross. And the reason it's a dihybrid, it's not just the two trait, it's not just any two trait, it's dihybrid because both individuals are heterozygote for both. So remember, when I say dihybrid, you have to be, each individual has to be heterozygote for both. Okay. If one individual generally looks like this, then it's not a hybrid. Okay, it's not a dihybrid. Okay. And so I asked this question here: what proportion, or in other words, what is the probability of the offspring expected to express both dominant traits? So this, I use the Y's and the R's like the P's we were looking at, where big Y is for yellow and R is for round. The little y is green and the little r is wrinkled, right? And so both for dominant traits, we're asking, what is the probability if we do this cross without doing the Punnett square? Okay, what is the probability of getting yellow, because that's the dominant, and round? And so in order to do this, we have to first figure out what are the possible genotypes for that. So these are, generally speaking, in order to be yellow, you need to have at least one of the dominant alleles. The same thing for round. So we're asking basically to get what's the probability of getting this. But now we need to write out what are the possible genotypes. So I'm going to go ahead and write those out. One way to do it is big Y, big Y, big R, big R. Another one is both heterozygote. Okay. Another is one homozygous dominant, the other one heterozygous. Okay. And then a final one would be where the opposite is the case. The first one is the first trait, we're heterozygote. First character, second character, we're homozygous dominant. These are the possible combinations. What are we going to do now is we're going to treat, because remember, when the events are independent of each other, it is a multiplication rule. Okay? And so what I want you to do is think back to your Punnett squares. And what you're going to do is what happens, you remember that what happens with one of the characters and their alleles is independent of what happens with the other one. That's the law of independent assortment. So there's the law of segregation where we separate the alleles. That's where you separate the individual alleles for each character. That's just segregating them. But the way the Y's segregate from the R's are independent of each other when they're on separate chromosomes, the two different ones, right? So if that's the case, if what happens with this one is independent of this one, then all we need to do is figure out the probabilities for each trait and then multiply them, because what happens with the Y trait is independent of what happens with the R trait. So we multiply those probabilities. So we think back, treat these like monohybrid crosses. So first focus on the Y's. And let's generalize here so I don't have to do them for both. Generalizing instead of using Y's or R's or A's. If we have a monohybrid cross, okay, for any trait, then we know we're going to have 1/4 chance of getting big A, homozygous dominant. We're going to have a 1/2 chance, or 2 out of 4, which is 1/2 chance of getting heterozygote, because both of these squares here, top right and bottom left, are heterozygote. What are the chances of getting homozygous recessive? Is 1/4. Okay. So now we think about that, then we're going to come over here and say, well, what are the chances? Because we're going to treat, this is our actual cross, the dihybrid. We're going to analyze each, the Y trait and the R trait. We're going to, or the color trait and the shape of the P separate. So let's look at this one first and say, well, what are the chances of getting homozygous dominant? We come back over here as if we were doing a monohybrid cross, and we'll say, well, that's 1/4 chance. Okay. Now remember, what happens with the Y's is independent of what happens with the R's, the shape of the P. And so we go over here and say, well, what are the chances of getting homozygous dominant? Again, we come back to this, the monohybrid cross, and it's a 1/4 chance. And because what happens with one is independent of the other, that's multiplication rule. So we multiply those two. 1/4 times 1/4 gives you 1/16, because 4 times 4 is 16, right? Now we're going to go about and do this with the others here, okay? And for what are the chances of getting heterozygote? The chances of getting a heterozygote, let's go ahead and keep, we could reduce it to 1/4, but let's keep it, there's 2 out of 4. This way we don't have to find common denominators later. Okay, so 2 out of 4 for a heterozygote for the first one. The second one is independent. That's going to be 2 out of 4. Because if we reduce it, it would be 1/2, 1/2. And when we do that, we're going to get 1/4 over here. And then because we're going to have to add them at the end. Because remember, an individual can only get one of these genotypes. An individual can't get 2 types of genotypes. So these are exclusive. So once we find these here at the end, then we add them and we apply the addition rule there at the end. Because an individual can either be this, or this, or this, or this one. It can have those 4 genotypes, but it can't have more than one of those. But if the individual has any of these, then they're going to be dominant for both. So there's where the addition rule comes in at the end. So continuing with this process here, we're going to get 4/16. And again, we could reduce this to 1/4 if we wanted, but let's keep it at 16. That way we have common denominators we can add at the end. Continuing with this, Y homozygous dominant here, that's 1/4 chance. And we're going to multiply that by heterozygote according to our general monohybrid cross square. That's going to be 2/4. And then here at the end here, the heterozygote is 2/4 chance. And being homozygous dominant is 1/2. Okay. Let's go ahead and do the multiplication here. This is going to give us 2/16. And then 2 times 1 on the top is 2, and 4 times 2 is, did I say, did we do, this one here should be homozygous, that should be 1/4. I don't know why I put 1/2. Okay, 2 times 1 is 2, 4, that's 16. Okay, since all of them have common denominator, we can add these, so we know that the bottom is going to be 16. Let's go ahead and add the numerators for each one. 1, that's 5, 6, 7, 8, 9. We get 9/16. Boy, doesn't that match that ratio we memorized? 9 to 3 to 3 to 1. What did the 9? The 9 was showing dominant for both when we were doing the dihybrid cross. So we get the same thing without doing the Punnett square. Me explaining it and going through it, actually, it took more time than it would. Doing it this way actually takes less time than trying to do a 4x4 Punnett square, to be quite honest with you. So I have another one here, and this one is a more complex one, and there's no way in heck you would want to do this with a Punnett square. This is a trihybrid cross. Well, we can apply the rules of probability here, just like we did for this dihybrid cross. We can do the same thing for the monohybrid cross, and just pay careful attention to what this question is asking. I asked a somewhat simple question from this. So here it says, what proportion of the offspring, if you have this mating that occurs here between the 2 trihybrids, what is the proportion expected to display the dominant trait for character A, but recessive for B and C? What does that mean? That means that the individual can be homozygous or heterozygous dominant, but for the other 2, and we're just generalizing here with A's, B's, and C's, We could have picked on peas and used traits like yellow pea, round, and tall, and then use those traits. But here, just generalizing. And it's going to be recessive for the other two. So this is it. The only difference can be right here in this position. So there's two possible genotypes here. What we need to do is list them and then go calculate the probabilities for each individual. So in order for us to fit this, we can have an individual that's homozygous dominant for the first one, but has to be recessive for the other two traits. The other possibility, the individual would show the dominant trait if they're heterozygote for that A character. And homozygous recessive for the other two. Now we go through and we're going to treat each one of these as independent of each other, and we treat this like individual monohybrid crosses. So just focus on the A's first, then look at the B's and so on, and we go back to our general monohybrid crossing. So what are the chances of being homozygous dominant according to that? Is 1/4 from the general monohybrid cross. What are the chances of being homozygous recessive? It's only that 1 out of 4, that bottom right box there. So that's times 1/4. And then again, another homozygous recessive times 1/4. By the way, guys, if you were to do a Punnett square for this, it would be an 8x8 Punnett square, so you would have to fill in 64 squares. Forget about it. It would take forever to try to answer this question. But if you use probability, you get the answer right away. So here we get 1. 4 times 4 is 16 times 4. 16 times 4, that gives you 64. Down here, we get a heterozygote. So heterozygote, remember, is 2 out of 4, 1/2, but let's keep it as 2 out of 4. And then times, for homozygous recessive, is the same as the one above. 1/4 times 1/4. So 2 times 1 times 1 is going to give 2, and 4 times 4 times 4 is 64. So what are the chances? The individual either has to be this one or this one. It can't be both, right? And if you have many offspring, then individuals that are going to fit this are either going to have this one or this one. So we apply the rule of addition here at the end. Common denominator is already there, 64. So we add the numerators, 2 and 1, that's 3. So the chances are 3/64. So I think it's about 4.5% or something like that, a chance of doing that. And here's your answer right here without having to do 64 squares of a Punnett square, an 8x8 overall. And that brings up one other item here, is being able to figure out, we also could use a multiplication rule to figure out how many possible gametes are produced here. And when we look at a heterozygote, okay, how many possible gametes? Well, because we're going to segregate, it's 1/2 chance of A, 1/2 chance of B, so there's 2 possible ones, so it's 2 possible gametes. But what happens if you have a dihybrid, right? Not dihybrid cross, but just a dihybrid, okay? Well, there's 2 possibilities here because you can either get a big A or a little a, and that's independent of this one. There's 2 possibilities here, big B or little b, so how many possible gametes is 4, okay? Now, what if I told you, so this would tell me that I would need a Punnett square that has 4 columns or 4 rows for that individual, right? So looking at a Punnett square, then, if we were to do a cross between a dihybrid for one individual and the other one, this is not going to be a dihybrid cross, though, because both individuals are not dihybrid. And let's say the other individual is like this. So how many squares would you need? Well, here we would need, for this first individual, we're going to need a 2 times 2, we're going to need 4. So you're going to need 4 columns because the first individual goes on the top. And what about here? Well, this individual can only produce 1 kind of gamete with A, right? But for B, it can produce 2 kinds, so it's 1 times 2. and so you would need 2 rows. So here you would need a 4x2 to do this, because this individual can only give one type of A. Okay. How many possible gametes then would there be for one of these trihybrids here? Okay. So I'm going to ask a question about this. It's just like this. How many possible gametes can be produced? It has nothing to do with figuring out fractions, just how many possibilities. Well, there's 2 possibilities, big A or a little a, right? The gamete also has to have either a big B or a little b. And it's equal chance of either one, but there's 2 possibilities. And the same thing here. So it's 2 times 2 times 2, and 2 times 2 is 4, times 2 is 8. So if you were going to run a Punnett square with this, you would need 8 columns for it. And so I might ask questions just like that. So in this section, we're going to be looking at what's called a Testcross. And this is going to help us determine genotypes for individuals that express the dominant phenotype. Phenotype's what it looks like. So we took something like in the peas, the flower color. Purple would be dominant, but you don't know if that individual has a homozygous dominant or a homozygous or a heterozygote genotype. Both of those would give you purple. Now, a white flower, we would know for sure its genotype based on prior crosses that have been done with pure breeding or true breeding plants. And so, what happens if you were given a plant that has purple flowers? How can you determine what its genotype is? And the same would be true for any other character that shows that simple dominance. Plant height, for example. Remember from an earlier section, tall plant was dominant to shorter plants. So both homozygous dominant and heterozygote, and you don't know what their actual genotype is. The answer is to do a test cross. So your learning objective here is to interpret data from a test cross to be able to infer what the genotype of the individual you were doing the test cross is. So the basic idea is that when an individual expresses genotype, you don't know a dominant phenotype. You don't know what its actual genotype is. So that second letter, regardless what parent you got it from, you don't know what it is, because all you need is one dominant allele, and you're expressing that situation. You're expressing the dominant phenotype. So let's say that we have an individual that you're given, and you don't know what it is. This could be a F2 generation, or just any purple pea plant. You don't know really what its genotype is. You don't know the history of it. So we don't know what that letter is. So the answer then would be to cross it with a recessive plant. And the recessive plant is known to be, the genotype is known already. Like I said, from prior analysis of data, we know that it would have to have homozygous recessive. So the idea here is you run this cross, and keep in mind, anytime you run a cross with these plants and you're looking at a character that's shown in the mature plant, you have to do the cross, let the new seeds be produced, and then plant them, and then watch them grow, and then see what you get. So let's say we do that. We do this test cross, and then we wait for this parent right here to produce the seed pods. And then we're going to get those seeds, and then see what we get. And we can actually predict what we would get in case where the individual plant that's expressing dominant is homozygous dominant. We can predict what we would get and then wait for the results to see what we get. Okay. So the prediction, if you're homozygous dominant, this individual can also be heterozygote. So we can see what the predictions would be. And right away you start to see that if the individual was homozygous dominant, that individual would be pretty much like the pure breeding crosses that were done by Mendel. All individuals would produce purple flowers in that next generation. Okay. If all you got was all purple flowers when you do this cross here, and you didn't see any white flowers, so you got to plant them and then wait for them to grow, the offspring of this test cross. So this is your test cross. And you got to wait until the plant you're doing the test cross with produces seed pods and from the pollination. And wait till you plant the seeds from those pods and then grow them and then let them mature and then look at their flowers. If all you get is purple, that would infer that the individual was homozygous dominant. However, in the other case, if the individual with unknown genotype was heterozygote, well, you're going to be getting, you should get some white flowers and some purple flowers. And not only that, you can even go as far as predicting the ratios. You can predict that 2 out of 4, or basically 1/2, would be purple, and 1/2 would be white. Or basically you would get a 1 purple to 1 white ratio. Whereas over here, you would get all purple. Okay. So if you got white flowers back, then that would infer that the individual was heterozygote. So that's the idea of a test cross here. This is the actual image that's given in your book, and it does so with illustrations. So it's pretty much the same thing I did, except they show the color of the flowers there. So we don't know. You're given a dominant phenotype, purple, and it can be either homozygous dominant or heterozygote. If it's homozygous dominant, then you're going to get all purple flowers. If it's heterozygote, then you're going to expect half are going to be purple and half are going to be white. Now we can also do a test cross with a 2-trait, looking at 2 traits. And we go back to our peas as an example here. Remember that yellow peas were dominant and round peas were dominant, the seeds from the peas, the pea pods, the things we eat are the seeds, right? And so it's possible that the second allele could be dominant as well for both, so there would be homozygous for both, and this would be the case here. And these are the other possibilities when you're given a yellow round seed. You don't know what it is, so you're going to want to do a test cross. In this case, you're going to test cross it where the alleles, where the genotype is homozygous recessive for both. Just like in a 1-trait test cross, you do just the homozygous recessive. Here you got to do it where the individual is going to be both green and wrinkled, okay? Because you know the genotype for sure. That's the genotype. So when you do that, you want to think about, well, what are the possible types of gametes you can get? Well, there's only little y to give, and there's only little r. In fact, we can do that. The first, the outer, the inner, and the last, and all you're going to get is little y, little r. So this is the only possible gametes that can be produced from here. So what does that mean? That means that you're only going to need 1 column. Well, this is the row here. You're only going to need 1 row because you don't have to put YR, 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 little y's and give 4 rows, because the ratio would end up reducing anyway from whatever ratio you get. Out of 16 to it'll reduce a number by a factor of 4. So you can eliminate it and still get the same ratio. And then here, so now we got to do what we expect if the individual was heterozygote for both, right? And so there we go and we figure out, well, what are the possible gametes? First, outer, inner, and last, right? And so that would be that you are the possible gametes for the individual that you're doing the test crosses. This is the individual there would be big Y, big R. Another possible gamete would be a big Y, little r, then a little y, big R, and then with the last 2 possible combination, you get recessive for both. Little y, little r. And so your Punnett square is reduced. You don't have to put 3 more rows because every row is going to be using the same little y, little r, and you're going to get basically 3 other rows that match the ones above, so it reduces the ratios reduce anyway. So going through here, we're going to get just looking at the phenotype. What would we expect from this if the individual we're doing the test cross was to be heterozygote for both? We're going to get big Y, That's a big Y, little y, so this, we know this is going to be yellow, and we're going to get big R, little r. So this is going to be yellow and round. And then we go big Y, little r, little r. Here this is going to be yellow and wrinkled. And then this one is going to be green and round. And then the last one is going to be green and wrinkled. So we're going to get those same phenotypes we were seeing earlier, only we're not getting a 9-3 to 3-1. We're getting a 1 out of 4 yellow round, 1 out of 4 yellow. In fact, we're getting the recombinants that we saw in the dihybrid cross, but we're getting them in a 1-to-1-to-1 ratio, so 1 out of 4, right? If we had done the other 3 rows, we would have gotten 4 out of 16, which reduces to 1 out of 4. And if we go 1 out of 4 and we represent that like the ratios, it's a 1-to-1-to-1. So if you were to do this test cross and you get roughly these 4 genotypes, including the recombinants, as a 1-to-1-to-1-to-1 ratio, then what would we expect? We would expect we can infer that the individual was heterozygote for both. You can go play around with this and figure out what you might get for these other 2. And if the individual was homozygous for both here, then you think about it, this individual, if the individual that you're looking at and you're doing the test cross with, you don't know what his genotype is, but if it was homozygous dominant for both, then you're only going to get one kind of phenotype, because this one only has a big Y to give and a big R to give. When you segregate them, it's only a big Y and a big R, and that means the individuals that are produced are all going to have at least one dominant allele, and that means, without even doing the Punnett square, is going to be all are going to be yellow and round. So if you get all yellow and round, then it would infer that this genotype is homozygous dominant for both, right? So, and then you can go and you can work these for this one. And you think about this, all of the offspring here are going to be yellow because there's only a capital Y to give here, but then half are going to be round and the other half are going to be wrinkled, right? So you would get a 1-to-1, or 1/2 are going to be they're all going to be yellow, half are going to be yellow and round, and the other half are going to be yellow and wrinkled. You can verify that with a Punnett square, but you can see right away all you get is one kind of color, but 2 possible. And then the opposite is true here. You can run the Punnett square, and in any one of these cases, the test cross would help you reveal or infer what the genotypes would be. So those are test crosses there. So here we're going to look at extensions of Mendel's model. And your learning outcomes here to describe how assumptions of Mendel's model result in an oversimplification. So when we talk about Mendel's model, we talk about some of those ratios that we've observed from crosses, like the monohybrid cross. You get a 3-to-1 dominant to recessive. If it's a dihybrid cross, you get that 9-3 to 3-to-1. And we're going to see how we don't always get that simple of an inheritance pattern. So we're going to look at that, and the second objective is to discuss genetic explanation for what's called continuous variation, and then explain the genetic basis for observed alterations of Mendelian ratios. Okay. Why do we get sometimes different than a 3-to-1 or a 9-3 to 3-to-1 for something like a dihybrid cross? So Mendel's model of inheritance has some assumptions. One is that each character or trait is controlled by a single gene. Okay, that's not always true. Height, skin color, for example, are controlled by more than one gene. And that each gene has only 2 alleles. That's also not true. Sometimes a gene has more than 1 allele, more than 2 alleles. And we'll see that with like blood types. Okay. And then also Mendel's inheritance patterns assumes that there's a clear dominant recessive relationship. Okay, so it's the reason like we got only purple flowers or white flowers, and purple was always dominant over white. Sometimes you're going to get something other than what you expect there, so there's not an actual clear relationship for dominant recessive. The fact of the matter is most of these assumptions are not met, Okay, genes usually have more than 2 alleles, and many of our traits are controlled by more than one gene. Okay, so the first one we're going to look at is polygenic inheritance, and polygenic means more than one gene. So here, like skin color, for example, or human height, multiple genes are involved in giving us the observed phenotype that we get for a trait. So in this case, the phenotype is the accumulation or the summing of the contribution from the multiple genes involved. Okay. And we might use human height, for example. This is the reason why we don't just have tall and short people. We have a range of heights. Okay. And so what we see here is that sometimes they call these quantitative traits, and these quantitative traits are going to have a continuous variation, not just tall and short, but a range in between. And they also are going to show what's called a normal distribution, which, by the way, how I do determine grades. I grade on a bell curve. If you were to graph human height here, and the taller over here, and the shorter are over here, the shortest ones, and here we put frequency, and then you go and you randomly sample a human population, and you start putting the heights down here, and if you get a repeat height, you start stacking them, you're going to start to get a pattern. Okay, so every individual that has the same height as another, you go and you stack them. Okay, so this is called a dot plot. And you end up getting few people at the end. This is frequency, right? So right here in this particular height here, we can see there's 1, 2, 3, 4 people have that height. So that's what's meant by frequency. And you end up getting this bell curve, this normal distribution, okay, with very exceptional short and exceptionally tall. And the average individuals are there in the middle. I grade very similarly here with your averages. I'll do a curve, and the middle of the curve there is the average grade. Exceptionally high grades over here, where the A's and the B's are, and then the lower grades out here. Okay, so the grades behave that way as well, but when it comes to inheritance patterns, this is what the normal distribution would look like. And here, instructor actually got students from their genetics class to go and stand based on their heights, and so on the left are the shortest ones, and so on. And this is a bar graph, called a special kind of graph called a histogram, and you can see you're getting roughly that bell curve, and so there's a range of heights. There's not just tall and not just short. And anytime you find a trait that has a range, a continuous range of distribution of the phenotype, in this case height, that would suggest it's polygenic inheritance. So that's how you would recognize. That's a big idea. I pulled another example to show you, not in your book, but skin color is another example. There are 3 genes that are involved in skin color associated with producing the pigment called melanin. And remember, for each gene, you have 2 copies, right? So there are actually 3 loci on your chromosomes, 3 locations where that code for producing melanin. So you can imagine if you have, for all 6 alleles, you have them all for coding for producing the pigment, and the alternative allele is for not making the pigment. Then if you were to inherit all 6 alleles, like you see over here, then you're going to get the darkest pigment. If you inherit none, then you're going to have the lightest color out here. And it's a continuous range, and you get this bell curve here. Here they're showing a cross between 2 individuals that are heterozygote for both, and they give the ratios here. When you do this cross, recall that it's 8 possible combinations for those gametes because they're heterozygote for each of those traits. That would be an 8 by 8 Punnett square, and so you would have 64 squares altogether. That's why those ratios are given out of 64 for each one. This is what you would expect to get for if you had a large number of individuals that had these particular genotypes producing it. If it comes to people, we don't have like 64 kids, but if you have many, many couples that have these exact genotypes, then we would expect these kinds of ratios on if we look at the larger number of all the offspring from there. Okay, so that's polygenic. And then we have pleiotropy, and pleiotropy is another phenomenon. And that refers to an allele that has more than one effect on phenotype. So it actually is responsible, the gene there, the allele is responsible for many other things. This is not surprising because gene products oftentimes are enzymes or proteins, and these products are involved in building different things in your body. Okay, and they may be the gene may code to produce a polypeptide that's an intermediate that goes through further change later, and that polypeptide can go through other changes as well, or the enzyme itself codes to produce a certain metabolite, right? And that metabolite can go through different kinds of metabolic pathways, right? So if that one gene is altered, then it can affect several different outcomes on there. So these effects are difficult to predict, and often they're not unknown what effects they are. They often reveal themselves when a mutation occurs, and in the disease you can see several effects in the body. So that one mutation that causes an allele to not produce the protein correctly, say an enzyme, can cause multiple phenotypic characteristics or symptoms of the genetic disorder. So they give examples there of cystic fibrosis that can cause multiple organs to shut down because of one faulty protein in the membrane. Another one is sickle cell anemia, and again, just one mutation in a gene creating an allele that essentially produces a defective protein can cause a number of problems. So that's pleiotropy. Again, often revealing itself when something goes wrong, right? Don't necessarily know what those alleles are responsible for doing in your body until something goes wrong. Then we have multiple alleles, and here multiple alleles, you have more than two alleles rather than just dominant and recessive. And an example of this would be with our ABO blood types in humans. There's three alleles. There's the type A allele, type B allele, and the type O allele. And these have to do with part of the surface covering of the blood cells, our red blood cells. It's covered with a glycocalyx, and that sugar covering that is there gives your blood a certain type to it. And it's important when it comes to blood transfusions and so on, it's genetically determined. And so this blood type, you have three alleles. And we're going to use an I to symbolize the allele, and then we're going to put a superscript indicating the type A allele. So an individual that was homozygous would look like that, right? So you have the I allele, the A allele, you have the B allele, and I'm going to explain more why I'm using I's in a little bit. And then you have a lowercase, which is going to suggest recessive for the type O. Okay, so this is the A allele, this would be the B allele, and this would be the O allele. And keep in mind, an individual can only inherit two alleles, right? You get one copy. An allele is a variation on a gene. You get two copies for every gene, one from each parent. You can only get two, but there's three three possibilities that you can get depending on what your parents give you, right, and what your parents were carrying in their genomes. Okay, so you can only have two alleles there, and so the fact that you can only inherit two, but there's usually more than two alleles for any given gene in a population, especially one that's outbreeding. And that's very important. And that means that you don't have a very small number of individuals that are constantly breeding with each other. There's a tendency for alleles to get lost, and then you start getting genetic defects associated with what's called inbreeding. There's too many close relatives related, whether it's plants or animals, that are breeding with each other. And the genetic variation tends to fade out. The alleles get lost from the population. That's not necessarily good. True for an outbreeding population tend to have more than two alleles, but the individuals can only inherit two alleles. So dominance, here's another situation here. We have multiple alleles, we have polygenic inheritance, we have pleiotropy, and these all represent things that are not easily explained by basic Mendelian inheritance patterns. There's another one here where dominance is not always complete. So we talked about there's a case where there's no clear dominant recessive, and one is called incomplete dominance. Okay, And here, the heterozygote is going to be intermediate, something in between. And so let's say that we had a red flower, pure breeding red flower of, say, a snapdragon flowering plant. And then we had a true breeding white, okay, flower. And you bred them. You did a cross. We don't know anything about them yet. We're just gonna, because the same species, just two different varieties of flowers, true breeding. So we know they're homozygous. We don't know which one's dominant, which one's recessive. And we're thinking, well, we're either going to get all red or all white. If this is simple dominance, and we don't know which one's dominant. But instead, we get all pink. And pink is intermediate between the two. This would be an example of incomplete dominance, okay, when it's a blending of the two. So now it might serve us well to not use capital and lowercase letters. Instead, we're going to use just a letter, like C for color, and then we're going to put a superscript of R for red. And so because these were pure breeding or true breeding, we're going to put that is homozygous for the red allele. Not dominant, just homozygous for it. There's not simple dominance. This is incomplete dominance. So over here, we're going to use C again, but we're going to put W for white. Okay. And if this is the case, remember the alleles get segregated into the offspring, so we only get to inherit one of these alleles, and there's only one type to get. So that means the first parent, the red plant, is going to give an R allele, and the other one is going to give a white allele, and the result is pink flowers. So there's your heterozygote. Now, so this is your parental population. This is your F1. Now, if we get the F1s to cross with each other to produce an F2, what would the expected ratios be? You're not going to get 3-to-1 dominant to recessive because you have pink in the mix now, right? So now we get all of these pink flowers and we cross those. So let's take a look at what the Punnett square would look like. Remember, we got to segregate the alleles into different gametes and into the offspring. The second parent, because we're breeding the F1s together, so we're going to breed two of these heterozygotes together. So the other parent would look just like that. And the first box is going to give us R, just like the parental. The second box on the top is going to give us pink. Okay. And then the bottom left is going to give us also pink. And then we're going to get white here. So what is the expected outcomes here? We're going to expect 1 red to 2 white. And I'm sorry, not white, pink. To 1 white. We could use fractions for this. We could say 1/4 red, or 25% red. 2/4, which is we can reduce to 1/2 pink, which is also 50%, or 0.5. And then we would expect the last box there is 1/4, or 25% white. Okay. So the expected phenotypic ratio would actually match their genotype ratios. Homozygous red, heterozygote for red and white, and homozygous for white. There's no dominant or recessive. This is incomplete dominance. This is what your book shows for this. Same thing we just did right now is that they have pictures here. We cross these. We get all pink. This is your F1, and then you cross F1s together. That's what they did there. And we get a 1 red to 2 pink to 1. Be able to solve incomplete dominance problems. So go through this one, work it out for yourself again, just like we worked out here, to see what you would expect. Okay. And then we have dominance. Co-dominance is different than incomplete dominance. And here, both types are equally expressed here. So there's flowers in the genus Camellia. There's a white camellia, red camellia. There's like hundreds of varieties. I think their origin is native to Asia, continent Asia over there. Hundreds of species there, but in some cases when you have some species that have red and white phenotypes and you cross them, you're trying to see which one's dominant, and then you get both red and white equally expressed. So you can see the hybrid right here. Okay, and when you see this, the flower is not pink, it's not a blending. You see red petals and you see pure white petals. This is codominance. Both alleles are equally expressed. So we might use the same symbols that we did before. We might say CW, CW, and then we might say CR, CR for those alleles. And then this one would be heterozygote. And we can go through all of the process here. And instead of half of them, when we run the Punnett square, are going to be pink, half are going to be flowers with red and white petals, right? So that would be your phenotype. So here it's not red, it's not white, it's not simple dominant, it's not pink. Incomplete dominance here is codominance, you get red and white. We also see this case with our blood types. Okay, and so for blood types, you can have different kinds of blood types. And that would take us to our next example on blood types for codominance. But we also want to cover, it's also related to multiple alleles. Okay, so here we have the ABO blood group. There's also positive and negative. That's a different gene, and we're going to ignore that for now. You can be positive RH or negative RH factor. And here we have a case of multiple alleles. That was mentioned earlier. There's 3 alleles, but we also have codominance. Okay, and that's why I gave capital letters to the A and the B earlier in the video, and a lowercase I because type A and type B alleles are dominant to I. So we have codominance for A and B alleles, right? So you're not going to be type A or B if you inherit A and B. You're going to be type AB. Okay, so the alleles are as I mentioned earlier, you're going to have the type A allele. We can use a superscript for that, a capital I for the B allele, and then a lowercase for O. So this is type A, type O, type B. And remember that these two are going to be dominant over the O. So let's take a look here. If you're going to be type A, what are the possible genotypes for type A? Okay, so we're going to make a little table here, genotypes. Remember, you have to inherit one from each parent. And if you know what to expect, then you can do genetics problems for blood type. So for blood type A, you can be blood type A if you are homozygous for the allele. And we don't say homozygous dominant because there's a codominance here. You can also be blood type A if you're heterozygote and you're carrying the O allele because type A is dominant over type O, right? So remember, this is type A allele. And this is the type B. And this is the type O. So that's how you can get type A. So you can be type A if you're homozygous for the A allele or if you're heterozygote with the A allele and the type O allele. For blood type B, you can be blood type B if you are homozygous for the B allele. Not homozygous dominant, just homozygous for it. If you're a heterozygote, type B, that would mean you're carrying one allele and it dominates over the O allele. And there's only one way to be type O. Okay. The way to be type O is if you inherit both O alleles. Okay, so the deal with blood type is an interesting thing in that you're already born with something called antibodies for your blood type. Usually antibodies, and so, because we're dealing with a virus right now, when you get sick or you get something that doesn't belong in your body recognizes it's foreign, doesn't belong there, and your white cells go to work to attack it, and then they start learning, they quote unquote, they learn about it, and they figure out by getting bits and pieces of the invader, could be like a bacteria or a virus, and they get bits and pieces of it, and then your body produces proteins called antibodies that go and stick specifically to that invader. And those proteins are called antibodies. Okay. Now you only produce them once you've been exposed to what's called an antigen. Okay. The antigen means stands for antigen generating or antibody generating particle. So antigen is a combination of antibody and generate. So antigens are anti-generating. So you get exposed to a bacteria or even an organ that doesn't belong in you, like during an organ transplant has to be perfect. If it doesn't have the right fingerprints, because that glycocalyx, the stuff you have on the surface of your cells are about your body recognizing itself. So anything that doesn't belong there, your body makes antibodies for this. But in this case, blood type is weird because our blood type, we already have antibodies for things we haven't been exposed to. Okay, so, and they use little symbols here to represent the antigens. The antigens are what's found on the surface of invading cells, or on the surface of our own cells. We have would be considered antigens to anything else. Like if any of my cells got into some other organism, some other mammal or other human even that doesn't have exactly the same genetic makeup as me, that would be considered foreign and an antigen. And so they show that for type A antigen, for blood type A, that A allele codes for a specific kind of surface glycoprotein that's there on the surface. Okay. Type B is another one, and they're symbolizing these with little circles for type A. They're not really circles. They're just symbolizing them for diagram, for understanding. Here they put these little cone-like structures for type B. And if you're type A and B, you'll have both. So you can see the type A, B. And then we didn't even put that. That's another blood type because they're codominant. So let's put that here, your type A, B. And you're like that. There's codominant. Can't believe I forgot that. And if you're that, you're both. You have both type A and B. Now, the deal about this is if you're one blood type, type A, you're going to have type B antibodies in you, even though you've never been exposed to type B. And so that is significant because if you bled too much and you needed a blood transfusion and you got blood from somebody who's type B, you're going to have the type B antigens, and that blood is going to go in there, and your blood plasma has antibodies, and they drew them with a little triangular shape, a Y shape right there to fit, because the molecules fit with each other. These antibodies are going to fit on those antigens and stick to them and cause blood cells to stick together, okay, and clump together. And that can be a problem because too many blood cells clumping together prevents the blood cells from flowing through small little capillary blood vessels, and you can't get oxygen to your organs. It can be very dangerous and deadly. The same is true for if you are blood type B, you're actually carrying A antibodies. So here they drew the antibodies differently. They drew them, the Y is a little bit different shaped. It looks a little round right there, kind of like lock and key, so that these fit perfectly and attach to the antigens on type A. So if you're type B and you get a blood transfusion from type A, you're going to end up getting that, what they call agglutination, and the cells stick together. So it matters what blood type you are. That's why it's important to know if you ever need a blood transfusion. And when you're donating blood, they want to know what blood type you are. Now, here's the funny thing. People who are both blood type A and blood type B do not have any antibodies circulating around, okay? And those that are blood type O don't have any of those surface glycoproteins there on the surface, and they actually have both antibodies. What does that mean here? That means if you're blood type O, you don't have any antigens on here, but if you get a blood transfusion from type A, type B, or type AB, you're going to have problems because you have antibodies for both of those antigens. So you cannot receive blood transfusions from any other blood type. It has to be type O. Okay? But because the blood cells themselves for type O don't have any antigens on their surface, you can give this blood to anybody else. To a type O, but you can give to anybody else. So type O is referred to as universal donor because the blood cells don't have any antigens on their surface to stick to any antibodies. So I can give this blood cells, when you give the blood transfusion, you give me blood cells, I can give this blood to a person who's type B, and those antibodies aren't going to stick to it. They're not going to cause agglutination. So, and then when we look at blood type AB here, they don't have any antibodies circulating around there, so they can get blood from anyone. So they're referred to as universal recipients. So if you're AB, you can get blood type A, blood type B, or type O, or your own kind of blood, and you don't have any of those antibodies circulating around, so no agglutination would occur. You wouldn't have any problems. Okay? So you should be able to solve some genetics problems knowing these definitions of these alleles. Okay. Now, your lab manual and some of the handouts I have available to you that are linked to your weekly assignment document do have some practice problems like this, and so do the study questions that I have. So make sure you look at those and then see about practicing with blood type genetics problems, right? For example, let's say you have a parent, two parents, and one is homozygous A, okay, and the other parent is type B but heterozygote, okay? So they would be like that. And I ask you, what is the outcomes? What are we expecting the children of these for their blood types, right? You should be able to answer that. You just do a Punnett square. Or I might just ask you one quick question. What is the chance? What are the probabilities? That means you can give percent or the fraction. How many out of 4 of the 4 boxes in the Punnett square? Is it 1 out of 4, 2 out of 4, which is 1/2, 3 out of 4, all of them? And ask, what are the chances of you getting blood type O here? And I can already tell the answer is 0, because the first parent is always going to give an A allele to all children, right? It only has A to give. And if you have an A and the other one, the other allele is type O, you're going to be type A. So there is no chance that any children would be type O in this for these parents at all. The probability is exactly 0. Okay, so they would have, and then I'm going to ask, well, what blood types would you get? Well, we would have blood type AB as a possibility. It doesn't mean that if they have one child, they're going to get AB. They can have 5 children and not have any type ABs. All their children could all be type A's. That's another possibility. And I go back to the coin example. When you flip a coin, you can flip a coin 10 times, and just by chance, random chance, you get all heads. Are you going to keep getting all heads if you flip it 1,000 times? No, eventually you start getting the law of large numbers, and the probabilities start to show themselves. The point is that we can predict what the chances are for any couple to have the blood types if you know their genotypes. But if you look at a large number of couples that have these exact genotypes having children, then we would expect 1/2 of them to be type AB and the other half to be type A in this particular case. This is just one example of a genetics problem. It will help you to learn the different types of genotypes that give you the different blood types. By the way, this is type O. Okay. Know the alleles. Know that A and B are codominant to each other, and you should be able to solve genetics problems because it's a simple Punnett square, a 2x2 Punnett square. Okay. And so this is just a chart from your own textbook that shows the blood types you get, the genotypes that give you those blood types, which we just went over there. It gives you the specific sugars associated with the blood types, which are part of the glycoproteins. And then it goes over who can donate, who can receive, which was covered in this other chart. I like this other one that I got here and I borrowed. Not exactly in your book because it shows an illustration of the blood cells. So what's in that table that you see on this slide is the same information that's on this one. I like the illustrated one better. Okay. There's also environmental influence on the way genes are expressed as well. There are some that are sensitive to, say, temperature, for example. Temperature is well known for determining sex, for example, in some reptiles, right? Depending on whether the eggs are at a higher temperature, lower temperature, you might get males or females, like in some turtle species, right? Well, here there's an enzyme that is part of the biochemical pathway for producing melanin, the color. And this is in some species of mammals, not in, say, in humans is not the case, but in some species of mammals, that enzyme called tyrosinase, which is part of the the molecule has to go through different changes. So there's several enzymes involved. And there's only one enzyme that does the conversion in one gene here. So it's nothing to do with whether you inherit multiple copies of a gene or whatever like you do in human skin color or hair color. This is different. There's an enzyme needed to make melanin to begin with, period. Okay. I believe it's called tyrosinase. Now, in the Siamese cat and Himalayan rabbits, that enzyme is actually sensitive to temperature. Okay? When the temperature is below 30 degrees Celsius, human body temperature, by the way, is 36 or 37 degrees Celsius. So in parts where the temperature of the body is below 30, and that's the case in the extremities, in the ears, the nose here, where the snout feels cooler, the tail, but where the core body is right here, that temperature is warmer or above 30 degrees Celsius, and the enzyme doesn't work above 30 degrees. So where the skin is a lot warmer, the cells that produce the hair fail to make the melanin, but where the body parts are cooler, the environmental influence of temperature causes the enzyme to work properly and produce the melanin there. Okay, so this is not true in all cats, but in a species of cat called Siamese cats and in Himalayan rabbits. And then we have a phenomenon called epistasis here. And here, epistasis has to do with metabolic pathways here. And in a metabolic or biochemical pathway, you have enzymes that are involved in making changes, and these enzymes are proteins made from a gene. And genes can come in more than one allele, right? So if a mutation occurs and then that allele is a faulty enzyme, it doesn't do what it's supposed to do, the mutation that caused the enzyme not to work properly, then you're not going to be able to get the product made. And so studying some corn, R.A. Emerson was able to observe this, and you end up getting a pattern that's not typical, 9-3 to 3-1, involving two different genes. So here, the two parental genes were white, and when he bred two white ones, and they were pure, true breeding, he ended up getting purple. Okay, purple-colored corn kernels in this variety of corn. So not true of all varieties of corn, but in this variety of corn here. So all of them were purple. And then when he got this generation, which is heterozygote for both. There's two genes involved. It turns out that this is the actual pathway. One of the genes codes for enzyme A, and that enzyme A causes a molecule that's going to eventually become that pigment. It's colorless. It causes it to go through a change, an intermediate that's still colorless, and this intermediate, which is different, but it's still not colored, that's not a pigment yet, doesn't absorb light and give a certain color, it goes through another change with the second enzyme. So both of these enzymes need to be working properly before we get the purple pigment. If one or the other does not work right, and you get both bad copies, then you're not going to get the purple pigment. And so that's the idea there for this epistasis here. And so when this experiment was run, where we crossed the F1 generation here, heterozygote for both characters, and you run that Punnett square the way we did, you get a 4x4 Punnett square, and you can see the ratio that he got was 1, 2, 3, 4, 5, 6, 7, 8, 9. He got 9 purple to 7 white, and this is not a 9-3 to 3-1, and the explanation is that you have more than one gene that's involved in expressing the trait that you're looking at, which is purple color here. So again, this idea of epistasis is seen in other examples, like Labrador retrievers. Those dogs, you have black labs, you have the yellow lab, and then you have the chocolate lab, right? Epistasis is seen there too. That's another good example they use there.